welcome to this top research seminar of medical genomics. My name is Heijmans. I'm scientific coordinator of the theme. And in the theme, we are passionate about fundamental research because we believe that it's the fundamental discoveries that we make in fundamental research that can revolutionize the treatment of diseases. And we also believe, well, we are sure that it's the technologies that we also have in-house in the LUMC, the state-of-the-art technologies that make all this possible. So today we have put together a program of various speakers like Suzanne Kloet, who will talk about the new technologies, Lucia Daxinger on one of the applications of those new technologies, Ahmed Mahfouz, again on an application and how it can link to a clinical application. And finally, Sylvie Noordermeer, also member of the management team of medical genomics, and Judith Kroep, who will together talk about how fundamental research can really link to the treatment of cancer. Our moderator today is Gijs. So he will monitor the questions that come online. He's also member of the management team of medical genomics. And afterwards, we will have drink and snacks downstairs, only in real life, not virtual. Susan, can I ask you forward for the first? Talk. Thanks.
Thank you, Bas. Good evening to everyone. So I'm very happy to be here to tell you more about who we are at the Leiden Genome Technology Center, as well as introduce some of the new technologies we've acquired this year. And hopefully you'll leave today a bit inspired about how you can use this technology to answer your own research questions. So first, for those of you who are unfamiliar with us, the Leiden Genome Technology Center, we're the genomics core facility of the LUMC, and we have three major focus areas. One of these is on single-cell and spatial transcriptomic technologies. And for this, we host several platforms from 10x Genomics, which is the market leader in this space. But we also offer several plate-based workflows for single-cell library preps using our Mantis platform. Another core area of support is on single molecule or long-read sequencing. For this, we have an Oxford Nanopore PromethION platform that I'll tell you more about in a bit. And we've also partnered with PacBio for more than a decade, and we currently have a Sequel IIe platform at the facility. We also offer several forms of custom support to LUMC researchers. This includes making Illumina preps that aren't available at a service provider. We have equipment available for DNA and RNA quality control. We serve as the central Sanger sequencing collection point for the Institute. And we're always happy to offer technical and protocol advice for users who are just getting started with sequencing or for people who need advice on which technology is most appropriate to help answer their research question. Now, because we're a core facility for the LUMC, we're available for all basic science and clinical researchers here at the Institute. I'm very happy to share that in 2022, we worked together with researchers from more than 20 different departments spread across all divisions of the LUMC. So you can see how important these genomic technologies are in many different research areas, and I hope we're able to continue this level of support for many years to come. Now, first, I'd like to highlight one of our new platforms in the single molecule sequencing space. So this is the Oxford Nanopore PromethION P24. This was an LUMC research investment that was installed at the LGTC in January of this year. And what the PromethION allows us to do is perform high throughput sequencing of any size DNA or RNA molecule. So this can be as small as cell-free DNA, all the way up to 2 megabase long segments of DNA. And why we really like the PromethION is it allows us to do on-demand sequencing. So each one of these 24 modules can be started and stopped independently. So this gives us incredible flexibility to do sequencing in the lab. And the platform also does what's known as real-time sequencing. So as the bases are being sequenced, we can monitor in real time what the sequences are, and that opens up a lot of new applications. So what I'd like to highlight is actually how the platform works, because I think the technical details are really important for understanding why there's so many potential applications. So at the core of Nanopore is the pore. So that's shown here in blue. And what we need to do is bind a motor protein, shown in purple, to the DNA. And the motor protein actually acts as a helicase, and it's going to unwind the DNA. And that's shown in the movie I'm about to start. Just a sec to make sure the sound is off. Good. So if you take a look here, you can see that this single molecule of DNA is being passed through the center of this pore. And as we pass through the pore, we're going to run a current. And what this allows us to do is actually measure the distortion in the current as the bases are passing through the pore. So what this looks like, I don't want the movie again, there. So what this looks like up close is shown here. So we call this the squiggle in the field because essentially we're looking at an electrical signal. And what we're measuring is the distortion or the change in amplitude of this signal as each individual base passes through the pore. Now, if you go back to your chemistry lessons, you'll remember that every base of DNA has a slightly different chemical structure. It has a different shape, and it's going to interact with the pore in a different way. So that signal is going to be different for all the bases, and it allows us to interpret which base was passing through at what time. So you don't have to do this one by one. Luckily, there's been algorithms that have been trained on millions of base pairs of sequence so that you're able to do this in a very high throughput manner. Now, not only can we look at just the A, T, C, and G of DNA, but Oxford Nanopore allows us to actually look at modified bases. So that's illustrated here. So again, we have the pore shown here, and we're passing a single strand of DNA through the middle. But in this time, we've actually got a modified base. So here's a modified cytosine. It's been methylated. And that methyl group is going to change the composition of the base. And this allows us to actually detect when the base has been modified because it's going to look different than an unmodified cytosine. So we can see very precisely which bases have been modified, which bases have not, and we keep the native sequence context. And this is something that Lucia is going to tell you about more in her talk about how this has been so powerful for the research that she's doing. Now, not only is Oxford Nanopore great at looking at methylated bases, but actually it can do a lot more. So we wondered what was available in the LUMC community. And the medical genomics team put out a call for Oxford Nanopore mini grants around this time last year. And we had 29 researchers submit applications for what they wanted to do with this powerful new technology. And in the end, we awarded 9 grants. And all of these researchers receive in-kind support from my team at the LGTC for the sample prep. And they also received support from Leon Mai's team at the Sequencing Analysis Support Corps, or SOSC, for the data analysis. So what I'd like to do is actually highlight the projects that we selected to give you all an idea about what's possible with the technology. So not only can we sequence DNA with the Oxford Nanopore, but we can also sequence RNA. And that's something that both Dongshu and Xuanan were very keen to do. So we can measure uracil, we can also measure modified RNA bases. So this is something that they'll be applying in their own projects. And another really powerful application of Oxford Nanopore is something called adaptive sampling. And this is something that Ronald Baus is going to look into on repeat expansion loci. And the way adaptive sampling works is because we're sequencing in real time, the sequencer can actually look and see if the sequence matches a region of interest. If it does match, then it keeps sequencing that molecule. But if it doesn't match, the pore can actually eject this strand and it can start sequencing a new one. So in this way, we're able to enrich on the fly for just our regions of interest in the genome. And we don't have to do any upfront amplification or capture. It's just happening directly on the sequencer. Now, long-read sequencing and Oxford Nanopore in general are very good at looking at full-length cDNA. So this is something that Noel de Miranda's group will do to look at the isoforms that are present in cancer samples to try to identify neoantigens. And Pietro Spitali's group is also taking this one step further, and they're combining spatial transcriptomic data together with isoform level resolution to see exactly which isoforms are expressed in which region of the brain. And finally, we had quite a few groups apply for traditional whole genome sequencing based approaches. So Viv Klaus-Smith's group will look at really small genomes, bacteria C. difficile. Marcel Teisterman's group is looking at structural variants in C. elegans and Arabidopsis. And then on the human side, Saskia Lesnick-Oberstein's group is interested in identifying causal variants from inherited cerebral small vessel diseases. And Pia Todenhaupt is looking at methylation differences between monozygotic twins. So you can see there's really a lot of applications that are possible with this very powerful sequencing technology. Now, in the second part of my talk, I want to highlight what's new in the single cell and spatial transcriptomic space. The first platform I want to mention is our latest acquisition at the LGTC. This is the 10x Genomics Xenium platform, and this was a Department of Human Genetics and LGTC co-investment, and it was installed at the end of June of this year. And why we're really excited about the Xenium is that it allows us to do in situ RNA profiling for hundreds of targets with subcellular resolution. So I don't have a lot of time to go into detail about how the Xenium works today. I just want to give you a quick snapshot of what's possible with this technology. So one of our recent runs was for a tumor microarray. This is from the group of Noel de Miranda. And we have 40 different tissue punches present on this slide. It's not to scale, but it's a 2.4 by 1.2 centimeter area. And all the colors that you see are actually the different cell types that were identified in this sample on the basis of the expression of 480 marker genes. So we're able to get subcellular resolution for 480 targets at one time. So it's a really powerful technology. I'd be very happy to talk to any of you if you're interested in applying this in your own research. Now, another big development in the single cell field is on the multiomics front. So when a lot of single cell protocols first came out, you could only look at one modality at a time. So just the DNA from a single cell, just the RNA, or just the protein. But recently, lots of groups have developed do-it-yourself methods to do multiomics, and there's also several commercial solutions that are available. So now we're able to combine multiple different modalities, and that gives us a much more nuanced view of what's going on in our sample. And that allows us to discover new cell types, get a better view of the heterogeneity present in the sample, discover new biomarkers, the list goes on and on. So one of the more popular multiomic applications that happens at the LGTC right now is using a technology called CiteSeq. And that allows us to combine both the transcriptome level information together with proteomic information. So I've got a quick illustrative example of how that works here. So on the left, you've got a hypothetical sample. Well, let's say you're interested in looking at the infiltrating leukocytes that are present in the sample. So already for 6 or 7 years now, it's been possible for us to get lots of independent single cell libraries from these samples. But what CiteSeq allows us to do is actually measure the abundance of epitopes in this sample. So that's highlighted here with our T cell, where we've got the TCR, but we also have an epitope of interest. And what we do is take barcoded antibodies. So these look a lot like antibodies you would use for flow cytometry, but instead of conjugating a fluorophore, we conjugate an RNA or a DNA barcode. And so when the antibody recognizes this epitope of interest, we're actually able to capture that barcode information at the same time that we capture the RNA information. So we get what we call a proteogenomic readout, where we can infer which proteins were present on the cell surface at the same time that we get the RNA measurement. And Ahmed's going to tell you more about how powerful this is in his talk. And I wanted to mention for the immunologists in the room, we can also get single cell B cell receptor and T cell receptor data at the same time. So possibilities are unlimited. So I hope I've inspired some of you to apply these new technologies. So you're always welcome to contact myself and my team. I've included our general email address here as well as my personal email address. So don't hesitate to reach out or talk to me over Bitterballen later tonight. And with that, I'd be happy to take any questions.
Are there any questions for Susan?
Yeah, I was wondering, you told about this adaptive sampling method, which seems pretty neat. Can you tell us a bit how much enrichment you can get? I mean, because yeah, it's a random sampling, so I guess you still need quite a lot of data.
Can everyone hear the question at home, or is it good that I repeat that? They can hear at home? Okay. Yeah, so with adaptive sampling, you've got flexibility to choose how large your target region of interest is. So the bigger you go, the less average coverage you will have. So there's kind of a trade-off there. But what we've seen recently with 120 megabase panel is that we get about 80x coverage within a day. So that's actually quite high, yeah. So now we're looking into opportunities to actually run multiple samples at a time and see how we can get this trade-off between throughput and coverage, yeah.
Any other question? I was wondering what the waiting time is. If you have a new customer, are all the machines fully booked, or is there room?
Machines are not fully booked, but my team sometimes is, as am I. So yeah, this time of year is very, very busy for us. But yeah.
People need to spend money.
So, yeah. And things always tend to wind down at the end of the year. So, but yeah, in general, we are doing our best to keep up with capacity. So you can always contact us, and you know, if it's really urgent, we push it through. So we're very flexible with that. Yeah.
Yes, in the Oxford Nanopore application, have you already looked at whether the DNA also contains damages that you can pick up by the current redistribution?
That's a good question. So we have not specifically looked into this, but often damaged DNA leads to fragmentation. So actually, we see a shorter than average read length,
But
and we
then-
think that they're breaking as they're going through the pore or as a result of the unzipping. Yeah.
But maybe base damages. So maybe they have, okay.
Yeah, entirely depends on the damage, but we have not specifically looked for this, but in theory, you could with the right base color. Yeah.
Okay, thanks.
Thank you, Susan. Now I can imagine, just a short intermezzo, Lucia. I can imagine that you come up with a question after the talks. And so over the last year, the medical genomics team, together with the IT department and the SASC, has been working on a forum for LUMC researchers, research Q&A. So the email address or the web address is there. So researchqa.lumc.nl. And when you go there, on a laptop or a computer that is within the LMC, you immediately log on to this forum, and you can use it as a regular forum and interact with other researchers. So there are multiple categories: research teams, research facilities, career development, Science Days Corner, and for example, here I made a message or follow up on this medical genomics top research seminar, and there we can continue the discussion afterwards if you were afraid to ask something or you just come up with something new. And also the speakers will be on the lookout so to answer your questions. So and also other questions to research facilities or whatsoever, please ask them there. This will, we expect, slowly become more and more used by, well, all researchers in the LMC, and it's accessible to everyone, to any theme and whatsoever, and it just works the way you are used to working with forums on the internet. So after this intermezzo, then there's Lucia Daxinger, who will talk about an application of the Oxford Nanopore technology.
It's on. Okay, yeah, it's on. Yes, thank you, Bas, and for the medical genomics, management team to give us the opportunity to present in this forum. And thanks everyone who turned up at this hour. And so my lab is really interested in epigenetic regulation and how epigenetic regulation influences, health and disease. And we are at the human genetics department. And throughout the past year or so, we've really been using Oxford Nanopore long-read sequencing to try to answer some of our research questions. And just, to bring everyone back to why why I care about DNA methylation and why it's just one of my favorite, chemical modifications to the DNA is, I think it's so nicely illustrated if we, yeah, this is this pointer. But anyway, if we look at this white little dot here, which is actually present in every female cell, and this is the bar body or the X chromosome, and really what epigenetics does, it allows a whole chromosome to compact into this tiny little dot, and then it stays there to be transcriptionally silent in every cells of our body. So it is really also for dosage compensation to keep a whole chromosome silent. And this is really also one of the main mechanisms of DNA methylation. The other thing I would like you to maybe remember is that it's a mitotically heritable modification. It's really crucial for normal development, and knockout or missense mutations in any of the enzymes that put methylation there, but also that remove DNA methylation, have been linked to human diseases. And the cool thing about methylation is also that it's actually reversible. So there's a whole machinery in your cells that is able to remove these methyl marks, and therefore it has, huge therapeutic potential if we were able to understand a lot more still about this mark. And so what what I would like to highlight today is really research that we've been doing in the context of human developmental diseases that actually exhibit or show altered DNA methylation patterns. And this is something because of with the advent of more genomics technologies and also improved methods to profile DNA methylation genome-wide, we have been able to just look at methylation levels in blood of many different patients with specific diseases. And basically what this, and this has been done by many research groups around the world, and this has led to so-called episignatures or DNA methylation differences between these disorders that are a very reliable and specific diagnostic tool in blood. And I will come to this, back to this at the end of my talk. And the other thing that we noticed from all these studies was that really many of these diseases, actually the disease-causing gene is also an epigenetic regulator. And this, to us, you know, has also is of biological interest in terms of like, if we have methylation changes, could they also link to phenotypic changes in these patients? But this has been a challenge in the field. So how can we link DNA methylation to disease phenotype? And one of the reasons why it is relatively difficult to do this, because one would have to do this in many different cell or tissue types, because epigenetic modifier genes are really, they show widespread expression across, you know, in all cells of our bodies. And if you have a germline variant in one of these modifiers, the aberrant methylation could actually be present in many tissues, in many cell types, but we really don't yet quite know what the consequences are and whether we would find the same consequences in every cell, or if it would be only in different specific organs, for instance. And we have decided to address this using a mouse model in a disease that actually shows quite a strong DNA methylation defect. And this is an ICF syndrome pathogenic variant, which is a developmental disorder that has phenotypes in the brain, but also in the immune system. And we have used a discovery-based epigenomics approach because this is really not well possible to do in humans. But what the mouse allows us to do is collect DNA, RNA, chromatin from different tissues at different developmental time points. And we can then use epigenomics technologies to simply profile DNA methylation, gene expression, chromatin, but also histone modifications. And what we set out to do was, you know, to figure out where in the genome in this mutant do the methylation changes occur, and where and when do these methylation changes translate to gene expression changes. And I will have to cut a very long story very, very short today, because it's about technology and how Oxford Nanopore helped us to figure out what is happening, or at least, you know, get us closer to an answer. Because when we looked at DNA methylation changes in the mutants, we did this using several approaches to measure DNA methylation. But what we always found very consistently across tissues was that our mutant showed about 40% DNA methylation loss compared to the wild type situation. And when we then looked more carefully where these methylation changes occurred in the genome, what we realized, or what we saw really, was that it affected groups of gene families. So basically, where you see there is a family of gene where the family members themselves are very similar to each other. And what this means still in 2023 is actually that we have gaps in our genome, because not all DNA sequences have been properly mapped, particularly at sites where a DNA sequence is very similar. And this is the case for gene clusters or for these large gene families. And so we have these sequence gaps in the genome, which means we also have very little information, first about DNA sequence, and second about epigenetic or DNA methylation changes. And this is a result because in the past we've all been using these short read and Illumina sequencing technologies, which means you have 150, or you have DNA sequence information about 150 base pairs in your genome. But if sites are very similar to each other, you cannot uniquely assign it to one site or the other. And what is happening is then that we basically are not sure what the information is. And really, with the advent of long read sequencing, I have to say we use both PacBio and Oxford Nanopore, but I'm going to focus on Oxford Nanopore today. so basically what it allows us to do is that with these very long reads, several kb up to, or what, yeah, 100 kb in length, we can actually cover a lot more DNA sequence and get more accurate information about DNA sequence, but also DNA methylation. So, and then really the question was, does this actually work, or is it just an empty promise? And does this allow us to now resolve these DNA sequence gaps that seem to be affected in our mutant situation? And if we use Oxford Nanopore technology, does this also allow us to get DNA methylation or accurate DNA methylation information about these sites? And then with, together with the LGTC and Susan and Mahfouz as well, we performed different PromethION experiments, and we did whole genome sequencing and also some adaptive sampling. But to start, just basically for the whole genome sequencing, what we used for our samples was about 600 nanograms of genomic DNA starting material, and we sequenced to up to 20 times genome coverage, because we are in the mouse, so it is a little bit easier to deal with DNA sequence. And I think this just, I wanted to show this because it shows very nicely that actually the average sequence length or molecule length that we get from the PromethION runs is 20 to 70 kb. So this is really, and we in the past we got 150 base pairs. So this makes a huge difference in terms of just what we get in sequence information, plus DNA methylation or epigenetic modification information from these types of experiments. And then the second one that was already introduced was adaptive sampling, where you basically, it's one of my favorite methods, because all you do is you sit on a computer, you decide on a genomic region, and then you tell this to the machine, please sequence this, and it actually does it. I don't have data today to show it, but I'm happy to talk afterwards about that. It really specifically enriches for the sites that you give the machine to enrich for, and the rest of the reads will be rejected. And so, but come ask afterwards, please. But really, so now, what does the data look like, and has it actually, you know, how well does it work for us? I want to just highlight 3 examples today, and one is basically that we were very much interested in what does DNA methylation look like if we observe allele-specific methylation patterns. And you may remember that we all have imprinted genes. And the imprinting happens because you inherit an allele from your mom or your dad, but you will express only one, whereas the other allele will get epigenetically silenced, and it does so by putting DNA methylation there. And if we look now at one of these imprinted loci, and we look at these reads, so these basically these green, you know, lines are the molecules. And then I think it's very nicely visible here that the first one, the top one, shows only these green dots, whereas in the lower one, you see these black dots. And if you just follow these reads, you will always see that there will be one molecule that is completely green, and the other one is completely black. And basically what it means is green is unmethylated, black is completely methylated. And if this, you know, if we sum up the reads, what we would expect, because it's one allele, you know, methylated, the other one unmethylated, it's 50% DNA methylation overall. If you then look at the data, this is really, you know, exactly what we see. And in this case, in the wild type and in the mutant situation, so we felt very comfortable with this technology that we can detect allele-specific methylation patterns. And the other example locus that I would like to show is basically single copy genes. And what I'd like you to, you know, take away from this is really that we have these different genes, and some of them use DNA methylation to at their promoters for transcriptional silencing. And this is really a specific feature of tissue specifically expressed single copy genes. And so one of the questions for us was also, are we able to pick up these heavily methylated promoters? And I'm just going to highlight here the Maelstrom gene. And we did this experiment in the brain. So, and Maelstrom is supposed to be expressed in the germline tissues. And what you can see is that this promoter region is really quite heavily methylated. And then the rest of the CpG island promoters are actually unmethylated in most of the tissue, and this was also true for most CpG island genes. And then, and so the locus next to it is such an example where you see that, that there's no DNA methylation detectable around the promoter of this ILDR2 gene. And if you then plot this again, I think it's very nicely, this completely unmethylated, whereas here we have a lot of DNA methylation. So this, looking at these control loci actually gave us, confidence in that we, are ready to go for these gene clusters or for these gene families. And so once we started looking at those, really the patterns that we see, and now we really look at the 1 megabase region, and basically, so all these genes that are here, this is a CIP class gene cluster, are very, very similar to each other. So we haven't had very reliable information about the DNA sequence, but also methylation state. But this, with these long-read technologies, this has really changed because we were able, to accurately map not only DNA, but we feel at least, improve DNA methylation mapping. There are a few gaps still here that if these like whiter sites where we have not enough read coverage. But the two things is basically, so we are able now to, map these unmappable sites. And what scientifically for us is even more exciting is that because in green is the wild type and in purple is the mutant situation, that you can really see when you go from single copy genes, where our mutant doesn't seem to affect DNA methylation. If you go to this gene cluster, it's a whole different story. And we see actually quite a specific DNA methylation defect at these clusters. And so from a biological perspective, we are of course super curious to figure out now what is happening in the mutant. But this, I'm not going to talk about this today, but what at the end I would just like to highlight one thing, because we are within the LUMC, we're a clinical department, also within the theme, we thought, okay, now we have this technology at hand. Can we go back to these neurodevelopmental or these developmental diseases and use this technology for a novel approach? And so what we did first was, to really look at these published methylation signatures and pick diseases that where the LUMC is an expertise center. And we interrogated more, not so much what is so different between these diseases, but where do these methylation changes actually occur in these disorders? And this was all done by old technology, but with the new technology, we can get a lot better resolution. And what we noticed there was actually that many of these DNA methylation changes occur in these regions where there are this gene cluster. So there is probably a lot more information that we can still gain from the diseases, in both in diagnostic, to improve diagnostics, but also to improve biological knowledge about these genes. And so what we've decided to do is that a group from clinical genetics, data science, and epigenetics, together with Susan's, that we teamed up, and we would like to address this in the context of neurodevelopmental disease. And really what the idea is to not only use long-read sequencing technologies to improve the blood episignature for diagnostics, but also see if we can translate this to a prenatal situation and use for novel diagnostic technologies. And with this, I would just like to really thank the people in the group, Maya, who has done all the biological work on this project, and Serkan, who really, was super enthusiastic, embraced all the Oxford Nanopore bioinformatics and the data analysis. And thank you for your attention.
Questions for Lucia? one thing that comes to mind is indeed so that we have this great technology that Susan can provide for us, but then you have the data. So can you say a bit more about what the challenges are once the data come off the machine and they sort of they bury you in?
They bury you.
Yeah.
Yeah. The data comes. The way we do it now is that Susan's or the LGTC, they do the base calling for us. So we basically, in bioinformatics terms, we get a BAM file, which means like we know the DNA sequence, but we also actually know the DNA methylation state. And this is something that we can put into our already established bioinformatics pipeline because we have been dealing with whole genome bisulfite sequencing data a lot. So for us, it was relatively easy to just adopt this for the nanopore, to be honest. But there is a very open nanopore community, and we've been in touch also with just developers to help us get code that was developed for human to adapt it to the mouse situation. So we've been in, I think there's more and more coming out, and everyone we have been in touch with, they have all been super helpful, and we're also happy to help everyone to set up their pipelines. Yeah.
That's great to hear.
Yeah.
Gijs, did I see you nod?
Yeah, I was just wondering about the biology. So is your idea then that this difference between single genes and these gene clusters, do you see that consistently?
Yeah, we see. Yeah.
Do you have any ideas how that?
Well, I cannot give you, like, we have not been able to do the proper experiment in a way, but yeah.
You mean no, I think.
No, I would love to know, I think the gene that is causing this very specifically affects this genomic compartment, but we do not have ChIP-seq data, as in I could tell you exactly where it anchors. Yeah.
Sylvie?
I can use this one, I think. Yes, it works. So you mentioned in the beginning that the good thing about methylation is that reversible and thereby it can be therapeutically targeted. But now you see that there's very locus-specific methylation differences. Are there any approaches that you know of that would also locus-specifically could revert this?
Yeah, that's a good question. So people have used CRISPR to target, and one of the more famous papers really is the FMR1 gene, where we know also what DNA methylation at the promoter does. So that is really used as a silencing mechanism. And then if you use DCAS to TET, you can remove it. And they have also done the ProBARDA experiment. They removed it and then put DCAS and DNMT3A, which is the methyltransferase. That puts the methyl group there. Yes, you can do it. Yeah.
Yeah, I also had a technical question. So I think the heterogeneity that you see in if you look at the locus-specific regions, so some sites are 100% methylated or at least cold, and some sites are maybe 80% or 60%. Is that biology, or is that the inability of the technology to have proper resolution?
I think it's both. It can be both, and this is and I think this also refers a little bit to what Bas was saying. I mean, we have a lot of additional data for these sites in terms of DNA methylation. So we use different assays. So we are fairly confident for most sites whether this is to say if this is biology or if this is a technological things. But I think for some sites, we probably the methylation calling is gonna improve, and maybe Susan can say more about this. But I think the methylation, also just the base calling for DNA methylation, is really something that is just getting better and better. But this it comes from Oxford Nanopore.
Okay.
Yeah.
Any online question, Gijs? No?
I think Anna has a question.
Oh, yeah.
Hey, it's very nice talk. Just as a comment, so recently we've been looking at methylation with short reads, unfortunately, but we found also, we've been focusing on microRNA clusters, and we see that they are fully methylated as well, except the promoter. So I was wondering if in these gene families, Do they share promoter? Do you see any signature of methylation going down? Because it could be the same mechanism than the one we have for these microRNA clusters.
They don't, it depends, well, you know, to be very honest, is like we don't know for every gene cluster yet what it really looks like. But for some of them, for some of the ones where we have looked a bit more carefully, we see that they don't share the promoter. For others, we simply don't know. So it's like, yeah, I think it's something to follow up. You know, maybe we should have coffee and look at the sites together.
Remco?
Yeah, does the Oxford Nanopore technology allow you to distinguish methyl cytosine from hydroxymethyl cytosine?
Yes, it does.
Okay.
And so I had to say that I've always been a bit hesitant to look at the hydroxymethyl calling because simply hydroxymethyl is not my expertise. So I'm a methyl cytosine person, but we have the data and I have now someone, who will help us in the end interpret it. But this may be also my personal hesitance a bit because I'm not sure what I get is if I will be, referring back to Marcel's question, is this now biology or is this technology? So we're trying to approach it together with someone who is really studying hydroxymethylation. Yeah.
Are you also seeing methylation of other bases than Cs in the CG? No, we haven't touched it.
No, but I know that. So the thing is, the non-CG methylation is really something that in mammals you find in brain, right? And there, the other thing, it's like it starts from around birth when the non-CG also seems to become relevant. And there is from mammalian systems, there's currently no pipeline, but the Arabidopsis people, they have a pipeline, and this is what I would like to adapt now for the mammals.
Nice. Well, thank you, Lucia. So we move on with our next speaker, Ahmed. Ooh. Oh, heb je deze of zo? You can point at this. Who will talk about functional genomics at single cell resolution.
All right, thanks, Bas. So indeed, in the group, we study a lot how cells basically change either throughout development or how things, if things go wrong, for instance, for particular cells, which sort of trajectory or path would these cells go through. So we, in general, talk about sort of perturbations that affect particular cells or cell types. And these can be either genetic perturbations due to genetic variation, can be environmental based on the environment where someone lives or sits, or it can be also due to a treatment that sort of pushes these cells in a particular direction. And our goal is to basically try to understand the processes that go on as cells go through these changes, with the aim that we can either, well, gain a better understanding, but eventually also be able to go back and reverse or arrest some of these changes. There is a big problem in the story that I just told you, is that we actually don't know our cells, right? I think if you open any textbook, you get an estimate of hundreds of cell types across an entire animal body. If you look at the recent science paper mapping all cell types in the human brain, you find more than 3,000 cell types, right? And it's debatable, of course, how functionally relevant those are, but there is a lot of work in trying to reconcile these transcriptionally defined cell types or populations with particular functions or other data, for instance, on connectivity in the brain, if that's the system that we talk about. So in general, I think that's sort of our aim in the lab, but many other people are also doing the same, where we try to build these atlases or maps of cell types in a particular system that we can then use to study basically how cells change according to particular perturbations. So to give you more of an example that we're working on at the moment is that we, together with a group of Maria Yazdanbach and parasitology, were interested in mapping immune cell states across different environments. And the environments that we're interested in here are basically whether people live in a rural or an urban setting. What they know already is that this influences a lot the composition of your immune system, and not only how your immune system is composed, but also how it will respond to certain challenges or interventions. Think about vaccination, for instance. So if you have a better understanding of this process, then you might be able to design better vaccines, for instance, rather than designing one vaccine that, for instance, works in European populations but completely fails in African individuals. So the approach we take there is that we collect PBMCs, so blood samples, and then we extract immune cells. And then we can either measure them at rest, sort of, or we can stimulate these cells with, in this case, we use vaccine adjuvants that activate different arms of the immune system. So Susan already told you a lot about CytSix, so that's the system that we have been using. I think, I'm not sure, but I think we have generated so far the largest data set that I know of in the hospital. So we have 200 samples. These come from 40 different individuals, and then we map them with these different conditions that I just mentioned. And if you extract roughly around 4,000 cells from each individual, then you end up with a data set of 800 cells. And the aim that we have now is to try to basically model this data to, again, understand changes in the composition across the different populations and how this affects the response to stimulation. I would also like to mention that since this is an ongoing line of research in the group of Maria, there is also more data from these samples. So we already have data coming from mass cytometry. We have measurements of glycans. We have measurements of metabolic enzymes and so on. So I don't have more results on this data to show you. I hope next time we can show you a little bit more on that. But I want to talk to you a bit more, because we're a computational biology group, is how do we actually try to model the data, right? So I think when you say model, that can mean many things. So I at least want to give you my idea of what that looks like. So in general, we deal with cells as points in a very high dimensional space, right? So for every cell, we have measurements of the entire transcriptome. So that can be roughly for humans, 20,000 genes. And this is a very high dimensional space. So things can look very messy in that space. But what we think, and I think there is a reasonable way to assume that, is that cells live in what we call a low dimensional manifold. So you don't actually need 20,000 dimensions to describe your cells, but you can go to a much smaller number that will give you a much clearer idea on how these cells are related to each other. And the reason for this assumption, why we think it's valid, is that genes don't work individually. They work in terms of programs or pathways. So then you can go to a much smaller space that can describe your cells. And that can be very informative. So in this cartoon that I show you over here, perhaps these gray cells are differentiating towards a particular path, and then some point they take an alternative route or something, right? And if we have this low dimensional representation of the cells, then it's much easier to model them or to try to represent this change in their transcriptional profile. So how do you do this? We borrow a lot of methods from machine learning and computer vision. So one of the methods we use a lot is called variational autoencoders. I will not explain it in details, but the idea there comes from the fact, again, that you have, let's say, an image over here, and that you want to learn basically let's see. Yeah, you want to learn here what we call a latent representation. So this is sort of a low dimensional representation of your image. Instead of using every pixel on your image to describe it, to show it, you can then say, well, okay, in this image, there is a person. They are on this point on the axis of a smile or having glasses on or whatever, right? So you can think of different attributes that you can use to describe this image in a probabilistic way. And then you train these neural networks. Here we call them an encoder or a decoder that try to take your image, squeeze it into these few attributes, and then try to reconstruct this image again. And if your network is very good, if it's trained well, then these attributes will be a very good representation of the image that we're talking about, right? So you give this network millions of examples, and it tries to learn where are you on this spectrum of having glasses or not, right? Or smiling or not smiling, yeah? Okay, so why is this relevant actually for single cell genomics? Well, we can do the same, right? So here I'm showing data that comes from different studies. So every dot is a cell, and it has a different color if it comes from a particular data set. And we show them in this UMAP space, which is, again, every dot, every two cells will be close by if their transcriptional profile is very similar. And what I want you to see here is that the colors are separate. So that means that cells group by study, and they don't necessarily group by the cell type, right? Which is not something that we particularly want. So we can now input these cells into these variational autoencoders, the same one that I showed you for the images. So we try to learn this Z over here. So this is again a latent representation of our cell that doesn't rely on the 20,000 genes, but let's say 20 dimensions or so that are a very good descriptor of our cell. And then we do another trick. We add this S over here that tells us which data set the cell comes from. And in this latent representation, we can sort of instruct the network to remove this effect, right? So I want to learn the relationship between the cells, regardless of which data set they come from. And if we do that, and then we try to use this Z to look at our cells again, what you see over here is that the colors are mixed. That means that this representation that I have learned of the cells is now free of this dataset effect. So now I want to show you an example why this is sort of relevant. So this is data set that Claudio in our lab collected and we published earlier this year together with Doreen Peters, where we basically collected all the mouse kidney single cell data that's out there. These are 8 data sets, a bit more than 140,000 cells. And again, we put these cells over here and we color them by cell type. And what I show you on the right side is how much these data sets basically agree in terms of the cell type annotation. I don't know if you have seen these upset R's plots before, but that basically means that all the data sets agree on one label. So all these people publish the data and then they give them different names, right? So we agree on one label. And that's not particular to kidney, by the way. If you look at any other system, that's always the case. What we actually want to have at the end is something like this, where we can build a tree that tells us what is the relation between cell types across all these different studies. So for instance, here we have a bunch of cell types that have this color code, and that means that they are really a perfect match or very consistent across all the studies. And on the other hand, we have these cell types here that have one color, and that basically means that we can only find it back in one of the studies that we included. So how do we do this? So again, we take all the data that we have, we feed it into this network. And once this network has been trained, we take this, again, space, which if you remember correctly, is free of these data set specific effects. And then we train something here that can reconstruct the tree I showed you before. I will not go into the details of how we do this, but it's a classification-based approach that tells us basically how cell type A in the first data set is related to cell type B in the second data set. I'll skip this part. So I want to show you an example of why do we want to basically match these cell types, right? It's nice in general if we want to build an atlas, but it can also have value downstream. So I guess most people are aware of genome-wide association studies, which basically checks the association of single nucleotide variants with a particular trait. And what you get out of these studies are these Manhattan plots, right? So for every location on your genome, you basically get a value that represents whether this location is associated with your trait or not, right? The problem with these is that most of these variants actually lie in the non-coding part of our genome, so interpretation is very difficult. So knowing basically why is this variant associated with my trait is a very difficult task. So what many people do is that they go to EQTL analysis, so quantitative trait loci analysis, where you try to check for a particular SNP if it's correlated with the expression of a nearby gene, right? So this is the example that you see here. You have a SNP and the gene, and if you plot basically for many individuals that have different alleles over here, if there is an association, right? So this will indicate that probably this SNP affects the trait through the change in expression of that particular gene. Now, these EQTLs have been used a lot. The problem is that we already know that these EQTLs are very cell type specific. So using bulk approaches, we could not resolve this before. So here is a very nice example from Monique van der Wijs from Groningen, where she basically showed that for this particular SNP and this particular gene, if you look at EQTLs in entire blood, you actually don't see any association. But once you break this into the different cell types, you see that in the CD4 T cells, there is a positive association, right? So probably the SNP is controlling the expression of the gene only in that particular cell type and not in other cell types. One problem with these EQTL analysis is that you need many individuals to be able to resolve this correlation accurately. In bulk, this is quite possible. So I think the cohort in Groningen now, they are up to 15,000 individuals or something that they are doing EQTL analysis in blood. In single cell, this is a bit more difficult. And that's why they have set up this single cell EQTL consortium, for which we take part in basically ensuring that all these cell type annotations are the same across the different cohorts. So at the moment, we are dealing with 16 cohorts that comprise around 2,000 individuals, from which we have genomic information, and we also have single cell measurements from blood. And this is a bit of old results that were taken only from 360 individuals, and then comparing basically the power to identify EQTLs to data from bulk that comes from 15,000 individuals. And what you see is that you actually have much higher power at the single cell level compared to the bulk data, even with fewer individuals, because you have this ability to resolve these EQTLs for particular cell types. So at the moment, we're doing the analysis for the 2,000 individuals. Okay, I was asked by Bas to show basically why is this relevant in terms of clinical practice. And I think the very honest answer is that these single cell technologies are very, very powerful in understanding the biological systems, understanding basically what's going on in a system beyond what we see in these bulk measurements. And there is a lot of promise in moving this into clinical applications. So you can immediately think about stratifying your patient groups, for instance, or looking into the effects of particular drugs on a subgroup of individuals and so on, and many other applications that are mentioned here. But I think the general challenges, at least if you look into literature, is basically first related to automation of your sample acquisition. So how do you automate the process of getting samples, performing the single cell experiments and getting your data, and then ensuring that you always have more or less the same quality. The same goes for the analysis, by the way. So I think most of the analysis at the moment is a lot of hands-on with back and forth between basically the data producers and the analysts. And I think this is something that needs to be automated if we are to move this to any clinical application. And the last one I put here is cost, but I think if we think about genome sequencing, Lucia today showed the whole genome sequencing that took the whole world 15 years, not so long ago. So I think this will go down pretty quickly. Okay, the last bit of my talk, I want to talk about the single cell network in the Netherlands, because a few years ago, we also realized that to be able to do this type of research, you actually need a lot of expertise. And that's why we have set up a network for researchers in the Netherlands that are doing single cell research. I believe the need for us came from two things. First of all, it's a very fast growing field. So on the left side here, you see number of publications and so on. So it is a hot topic. But as I said, it's really very highly interdisciplinary. So you do need people who know basically how to get high quality samples. You need people who know how to analyze the data. And of course, you do need the question that comes at the end. So that's why we felt a couple of years ago, and this started actually in Leiden here with many of the colleagues here. So with Susan, Anna, with Erik van den Akker and Wilde Miranda as well, that yeah, we do need to bring people together a little bit. So it becomes a little bit easier to do this type of work. So we started in April 21. This was called Single Cell Network Leiden. Now it's called Netherlands. At the moment, we actually have 400 members, and that spans really all the academic institutes in the Netherlands, but also many people from companies that are interested in the use of this technology. What we do is that we do a monthly online seminar. This started during COVID, but we still keep it because it allows us to easily invite people from all over the world to share their work with us. We did hold one physical meeting, and the idea is to make this annual. So the first one was here at the LUMC earlier this year, where we had more than 200 people in this hall. And I think this was really nice. And what we also did is that we tried to connect a little bit more to industrial partners who might not be using the technology at the moment, but certainly very interested. So we hold one event together with the Leiden Bioscience Park last year. which was very well attended with mainly people from companies, actually. So if you want to be in touch or involved, please look at our website. We make announcements there, and then you can join our mailing list as well. The next online meeting is in 2 weeks, where we have Ramon, who used to be in Barcelona. He will talk about a very comprehensive tonsil atlas, a human tonsil atlas. And our next annual meeting next year, and we would rotate across institutes. So we're going to Princess Maxima in Utrecht. Yeah, so please keep an eye on that. And yeah, with that, I would like to thank you.
Thank you, Ahmed.
Yeah, I'd like to ask a philosophical question. What's your definition of cell type? Because, well, as you show, I think there's lots of transitional states. So what definition do you use, or what is used in the field?
Yeah, I think it's a good question. It's also a very active question, I think, in the community. I think this is pretty hard. As in, if you only look at transcriptomics, I don't think you can answer this question, because you can always cluster your cells or group them in a different way, right? It depends on the level of granularity that you are interested in. To be honest, when you write publications, we never write a cell type. You always talk about a cell population, because that's I define this population, I'm interested in it, and then I do something with it. If you have complementary data, that helps a lot, right? So I think the example I mentioned about the human brain cells, they did not verify or check every single population of the ones they described. But for the few that they did, they could actually show either a distinct function or a connectivity pattern, or you have to show something that sort of distinguish this population that you describe from the rest. In my view, as we keep sequencing, I think it's just a full space, right? It's really up to you. We are now undersampling all the systems. In every system that we're looking at, we're definitely undersampling. So that's why you see these isolated clusters. At some point, you see a continuum, and it's really up to you to draw a border around, well, okay, this group I find now very interesting because they have X and Y properties, right? But I think the transcriptional space will be a very continuous.
It reminds me of your cartoon where you blame geneticists for just throwing all the genes over the wall to the neurobiologist, but you're just throwing cells to the neurobiologist. Do you feel guilty?
Well, no, I'm analyzing the data. I don't think. No, but I think it's a very fair point. The only thing is that now you're talking about transcriptional signatures, which are a little bit easier to link to function than if you just throw an RS code. Right.
Any other question?
Question about the dimensions. Do you have the model decide how many dimensions you need to? Yeah.
Yeah, think about it a little bit as principal component analysis. There you also have a bit the intuition that every dimension encodes a certain percentage of the variance in your data. With these deep neural networks, you don't get that immediately. So that is something that you need to play around with. But there are ways to optimize, like what's the optimal number of dimensions that I need for a particular task.
And what do you usually get? Can you say something about that?
It really depends on the task that you're working with. If you think about the defaults in the single cell field, for instance, I think it's 20 dimensions, but it doesn't mean anything. Yeah.
Coming back to the question, because you say, well, it will be in the end a continuum, but I think for quantitative measurements, what we do now is you have the clusters, and then you do a pseudo bulk analysis of a cluster to get to the expression QTLs, for example. So, I mean, that becomes problematic if there's no border in a way.
Well, you can also do sort of cell state QTLs. Mm-hmm. So do I have a variant that basically controls or associates with where the cell is on this continuum? So you don't necessarily need to rely on a discretization. Mm-hmm. I think discretization is still valuable if you want to, again, maybe go back to the clinic, because we will not be doing single cell in the clinic, I think. I think what you end up doing is that you define a particular feature or a variable that you can measure much more efficiently in larger populations quickly. And their discretization is still important.
Thank you. If there are no further questions. Thank you, Ahmed. So we continue with our last combined talk. We have a little fight with the pointer. Okay. That's an expert. So Sylvie Noordermeer is a more, I can say, more fundamental scientist who will be telling about her work, and that she will do together with Judith Kroep, who is a clinician and who is looking for an application of the kind of work that Sylvie and her group are doing in the lab.
Yes, thank you, Bas. This is indeed a dual presentation from me and Judith. And I think what we want to show you today is how fundamental research actually in the beginning came up with a novel therapy for patients that is now very well used in the clinic. But in the clinic, again, led to certain problems that we are now taking back in the lab to hopefully come up with better solutions even. So the talk that we want to give is not so much on a technology based, but more really on a clinical problem. And that's all around patients with BRCA1 or BRCA2 mutations, which I'm sure you've all heard of. That these mutations arise in families, which then have a very high probability of getting either breast and ovarian cancer in their lifetime. But with the recent genomics technology, we also now know that it is not only BRCA1 mutations don't only arise in genetic tumors, but also in sporadic tumors of different origins. So not only ovarian and breast cancer, but also, for example, lung cancer, prostate cancer, etc. And one very important characteristic of these cells is, and I see that I made a mistake here, is that these tumors are very genomically unstable. So the labeling is wrong. This is BRCA1 wild type, where you see hardly any genomic instability. And these are two examples of breast cancers with a BRCA1 mutation, where you see a lot of genomic instability going on in these tumors. And that is a very important characteristic of BRCA1 or BRCA2 mutated tumors. And that we can explain by the important function of these two proteins during a process of repairing double-stranded breaks in our DNA. You can imagine that a double-strand break in the DNA, which really disrupts the full helix, can be very disruptive if it's not repaired properly, can lead to translocations, deletions, etc. And luckily, our healthy cells have very good ways of repairing these via 4 different pathways to repair double-stranded breaks. And BRCA1 and BRCA2 are very important for this very fateful type of repair, which is called homologous recombination. And so in a tumor where BRCA1 or BRCA2 is mutated, we lose the capacity of cells to repair these double-stranded breaks in a very fateful way, which can lead to mutations, translocations, deletions that can give rise to tumor formation. And we also know that it is not only the function of BRCA1 and BRCA2 in homologous recombination, but also in 2 other very important processes that maintain genome stability. And that's why if we lose BRCA1, BRCA2, we get genome instability. So that's of course bad for the patients because that give rise to tumor formation. But in 2005, some very smart people also realized that this genomic instability can actually also provide an opportunity for treatment. And so that came really from fundamental researchers that thought, like, if we can really push double-strand break formation in these cells, maybe the double-strand breaks are so abundant that the cells that lack homologous recombination cannot overcome this and eventually will die. And that's exactly what they came up with. So they used PARP inhibitors, which are inhibitors of 2 important proteins that are involved in the repair of single-stranded DNAs. And And so if the single-stranded DNA breaks cannot be repaired by PARP inhibitors, this can give rise to double-stranded breaks. And in a normal cell, in a healthy cell that is BRCA1, BRCA2 proficient, so homologous recombination proficient, these double-stranded breaks will be repaired via HR and will lead to cell survival. However, in BRCA1, BRCA2 deficient setting, this abundance of double-stranded breaks is so much that the cells cannot cope with this and they actually die. This is a concept that we call synthetic lethality, which you might have heard of already. And that is a therapeutic approach that is now very of interest to a lot of tumors, actually. And the BRCA1, BRCA2 mutation tumors were really a driving force in this concept in the clinical fields. So with that, I would like to give the word to Judith, a medical oncologist here in the LUMC, that can give us some examples of how PARP inhibitors changed the treatment of these patients.
Yes, thank you for the invitation to give you a view in which we are standing in the clinical setting. Important for this translational research. Over here you see the survival data of various tumor types. And we know that BRCA mutations are happening in various solid tumors. Most often we know them from ovarian cancer and breast cancer. And as you can see here, an EKNL slide that shows the cancer-related survival. And you can see that the patients with the breast cancer, they do relatively well with a 5 years overall survival, about 86%. However, it's a large patient group, so there are still many patients who are in the palliative setting. While for the ovarian cancer, there is a high unmet need. Those patients have a 5 years overall survival of about 40%. And like Sylvie already told, the PARP inhibition has been developed and actually has been registered since 2014. And over here you see the timeline for the ovarian cancer. In the beginning, it was registered in the recurrent setting, so when the disease came back in a palliative setting. And since 2019, it's also registered in the first line, in the curative setting, therapy. And there have been studies from different pharmas with different PARP inhibitors. And olaparib mostly has been studied in patients with BRCA mutations, so the germline or the somatic mutations. And niraparib and rucaparib, they had large phase 3 trials in all comers. So in patients who are BRCA mutated or the HRD or the HRP, the HR proficient patients. And in the Netherlands, we from the Netherlands Dutch oncologists, we have our commissie CBOM. And all drugs that are registered by the EMA or FDA, they are also assessed by the CBOM, and they look at the benefit and cost, the real cost, but also the toxicity for the patients. And they look at the hazard ratio of the trial and also the benefit in time of months. And recently, those approval assessments has been tightened so that the guidelines are more strict, so that we can give, we are more selective when which medicine we can give to the patients. So to show a nice example about the PARP inhibition in ovarian cancer is for example, the SOLO-1 trial. It's a first-line maintenance study with olaparib in patients with stage 3-4 high-grade ovarian cancer with a BRCA mutation. And as you can see here on the x-axis the timeline and on the y-axis the progression-free survival. And then the red lines are the patients treated with olaparib. And then there is a medium progression-free survival benefit of about 3.5 years, which is really a lot. And it also gives an overall survival benefit. Another example also in the first line is the PRIMA trial with niraparib, so another PARP inhibitor. And this was tested in the all-comers. They also showed a nice benefit of the PARP inhibition. And what's nice about this trial that besides the BRCA-mutated patients, they also looked at the HRD group and even the HRP group. And what we see is that the PARP inhibitors, and it's also shown in other trials, work best in patients with a BRCA mutation, also have a good effect in patients which are HRD. But even in the HRP patients, there was a small significant benefit of about 3 months. And we do not understand it. We think, or the drug does more than we know. So it's more than only the synthetic lethality, or the test that we do is not good enough. So we are also currently testing with the lab of Maaike Vreeswijk that we look at the functional HRD of the RAD51 assay, that we treated patients with ovarian cancer with recurrent disease with only olaparib to see if a functional test might be better. And at this moment, there is internationally no standard HRD test. So internationally, but also in the Netherlands, research is ongoing for that. And of course, if you add a drug, it's added after standard chemotherapy. It can also give extra toxicity. In general, PARP inhibitors, it's an oral therapy. It's well tolerated. It can give toxicity, and then mostly it's fatigue or nausea. It can give some bone marrow depression. And over here you see the various kinds of PARP inhibitors that we can use for the different indications. What's also interesting is PARP inhibitors are also registered for breast cancer. We use them already in a metastatic setting, and they come soon also in the adjuvant setting. But the benefit is less than we see in the ovarian cancer. 2 trials and drugs that are registered in the metastatic setting for breast cancer are olaparib and talazoparib, and both drugs gave a median progression-free survival benefit of 3 months. So not 3 years, but 3 months, with a nice hazard ratio below the 0.6, but no overall survival benefit. And over here you see the timeline also in pink in 2017 that they were registered in the metastatic setting and recently in the adjuvant setting. So what I like to show is that PARP inhibitors, it's very nice that they work with the BRCA and inhibition of the PARP, that they can give synthetic lethality. We see a large benefit in ovarian cancer, less in breast cancer. It might be the difference of the locations of the mutations, that might be a reason. Also, research is ongoing in that topic. But still, there are patients who get recurrent disease, also in ovarian cancer. 70% of patients have recurrent disease, so there's still PARP resistance. And there's an unmet need for improved precision therapy and new, better agents. And yeah, then I've give the word back to Sylvie.
Yeah, and that's where we come in then again. And we tried, obviously, to find other additional, maybe better treatment therapies for these patients. So the way we set up this was by doing a genome-wide CRISPR screen in BRCA1 deficient cells. So we focused mainly on BRCA1 deficiency for this project, where we basically in the genome-wide CRISPR screen, we individually deplete every single protein of a cell in the cell and see the effects on the survival of BRCA1 deficient cells. So to look for novel synthetic lethal interactors of BRCA1. And when we did this genome-wide screen, we, for example, found PARP1, so that was a very nice confirmation. But we also found 2 proteins that really attracted our attention immediately, which were EXO1 and BLOOM. And these are 2 proteins that are involved in a very important step during double-strand break repair, namely resection. And that is the process where the double-stranded DNA surrounding the break is chewed away and then resolving a single-strand DNA stretch. And for homologous recombination, that single-stranded DNA stretch is very important to look for homology in, for example, the sister chromatid that can then be used as a template for repair. And these two proteins that we know play a role in this step of long-range resection. So you have short-range resection by MR11 and CTIP, and then long-range resection to really expose long stretches of single-stranded DNA. And if we compare this to proteins involved in short-range resection, for example, MR11 and CTIP, we see that there is not this unique BRCA1 specific synthetic lethality. So to read this plot, the higher the base factor in this case, that's the readout of the screen, the more essential the gene is. And you see that AXO1 and BLOOM are very essential for these blue lines, which are the BRCA1 deficient cells and not for the parental cells. So AXO1 and BLOOM really were very of much interest to us. So I'm a fundamental biologist. I wanted to know what is going on with these cells if they lack both BRCA1 and AXO1. So we looked at some phenotypical characterization of these cells. And the first thing that we observed was that these cells have very many so-called micronuclei. So these are the normal nuclei, so the happy stain, but we saw many of these micronuclei, and this is a measurement of genomic instability. Also, what we saw was that there's many chromosomal aberrations, like these kind of radials that you can also see over here, where different chromosomes fused, and also breaks in chromosomes, so really genomic instability of these cells, which is something that we already know from BRCA1 deficiency, but apparently AXO1 depletion even exaggerates this process. And that's when you also quantify this, you see indeed a very much increase of micronuclei and genomic aberrations. Interestingly, this was very specific for BRCA1 deficiency and not BRCA2 deficiency. So here again, you see the synthetic lethality. So in cells that are depleted for both AXO1 and BRCA1, you see hardly any cell survival. But if we did the same experiment in BRCA2 deficient cells, here in green, we actually don't see a very much increase of cell toxicity when we deplete AXO1 over the loss of BRCA1 by itself. And also, we didn't see this increased genomic instability. So really pointing to us that AXO1 loss is a very specific essentiality of BRCA1 deficient cells, which would be then very important for more precision-based medicines. So we wanted to more mechanistically understand this synthetic lethality. And we know that chromosomal aberrations, micronuclei are often formed because of a problem with double-strand break repair. So that was the first thing that we looked at to see whether these cells suffer from high levels of double-stranded breaks. And we do that by looking at the phosphorylation of HOX, and that's a very specific marker of double-stranded breaks. And indeed, we see that in the BRCA1 deficient settings, we see a huge increase of gamma HOX formation in the setting where also AXO1 is lost. And indeed, we don't see this in a BRCA2 deficient setting. So indeed showing that the toxicity is linked to double-strand break formation. So then we went back to our model of how double-strand breaks can be repaired. I already mentioned, so BRCA1 and BRCA2 are very important for homologous recombination. But there are actually two other pathways, alternative end joining and single-strand annealing, that are also dependent on this step of resection that AXO1 is mediating. So the generation of single-stranded DNA. And particularly single-strand annealing is dependent on this long-range end resection that is mediated by AXO1. So obviously our attention went to this process and how AXO1 will be involved in that. So I won't go into the technical details. We have reporter systems in the lab where we can measure the activity of these different pathways. And indeed, what we noticed is, as we expected, the depletion of BRCA1 or BRCA2 has an effect on homologous recombination. And actually, this effect is not exaggerated by loss of AXO1. But if we look at SSA, we see that already in a AXO1 proficient setting, BRCA1 deficient cells have a problem with SSA compared to wild type cells, while BRCA2 show the opposite and actually have an increased level of SSA. And then if we deplete AXO1 on top of this, we see that it doesn't affect the BRCA2 deficient cells, but it even reduces SSA even more in the BRCA1 deficient setting. So that really pointed to us that the role of AXO1 in SSA is the essential role for cell survival in BRCA1 deficient cells. So to make this a bit more clinically relevant, or to see whether this is clinically relevant, we looked at patient cohorts, expression data, where we see that, so these are 2 cohorts of breast cancers, that we see that breast cancers with a BRCA1 mutation actually show elevated levels of AXA1, which points to an essentiality of these genes. Like you can imagine that if a protein is very essential for a cell, it might actually upregulate its expression, and that's exactly what we see here. And that's also something that is known for other synthetic lethal interactors. And again, we don't see this in BRCA2 deficient tumors. So really pointing that this AXA1 is also in clinical settings a relevant target for BRCA1 deficient tumors. So that brought us to this mechanistic model. So what I showed is that BRCA1 deficient cells are very dependent on AXA1, and that is because of a defect in both SSA and homologous recombination. So this fundamentally is very interesting, but obviously also gives rise to therapeutic options. We could maybe think of AXA1 inhibitors in a clinical setting to treat BRCA1 mutated tumors. So that is exactly what we hope to do. However, there is no AXA1 inhibitor. So that's where Timo Wendel, a postdoc in my lab, comes in, who is gonna look for these AXA1 inhibitors. So the way we set up to do that is by setting up an in vitro assay to measure AXA1 activity. So this is a biochemical assay, and this assay we use in a drug screen. So this is a screen done with 6,000 or around 6,000 compounds, which is a drug repurposing library. So compounds that some are somewhere already in the preclinical development. So either in a phase 3 trial already, or launched even, or in a preclinical discovery. And we tested these 6,000 compounds for their activity to inhibit AXA1, and everything that is in red here are actually compounds that show some inhibitory effects. So obviously we had to downsize that list. And with some cherry picking, we eventually came up with about 6 hits that we indeed also in dedicated experiments could show really have an inhibitory effect on AXA1 activity. And at the moment, we're mainly focusing on this first hit, where we can actually show that it has toxicity in a BRCA1 specific background. So in dark yellowish here, you see the BRCA1 deficient cells, which are most sensitive to this compound compared to BRCA1 proficient cells or AXA1 depleted cells. Obviously, we're far from clinical approval of these compounds. The people that are having a close look on our data see that the IC50 of this compound is quite high. So we will need to work on the potency of this inhibitor and also on the specificity. Data that I didn't show, but we know that AXA1 has very close family members that are also inhibited by this inhibitor. And for that, we are working hard on a public-private partnership to have some industrial partners help us with the chemical optimization of this compound. And obviously, we've done many experiments in cell line models, but we now also need to go to animal models and also study potential for monotherapy or combination therapy, for example, in combination with PARP inhibitors. So that brings me to the conclusion. So the mechanistic work was done mainly by Anne in my lab and Bert van der Kooi in Heiko van Ettikum's lab. And Timo is very important for this drug development project in the lab. And of course, I also like to thank Judith for giving the clinical insights on this topic.
Questions for Judith? For Sylvie? Please. I guess. Yeah, Marcel first, someone else. Yeah, this. Yeah,
I just questions about your drug. So it is also always attractive to start with a repurposing thing. And it's a bit silly because then you find something that's actually meant to target something else. You find a modest activity, you have to change it. So my question actually, why don't you just dump it in the bin, start anew with a proper screen with 1 million compounds? There's plenty of options you can choose from.
Yeah.
And that will give you the freedom, because you'll be, if you focus on this one, you get locked in for another 2 years and you're still not going anywhere.
Yeah, no, I think this is a very valid point. That's also definitely something that we want to pursue. So actually the first hit that I showed it has multiple reasons why we are interested in. So we know it's very similar to a drug that's already described for an exon 1 like protein. So that it really shows us more data than just the screen data. But we definitely think that also other compound libraries should be done. So yes, this is something that we also want to pursue. Yeah.
Judith, so you said that PARP inhibitors work much better on ovarian cancer than breast cancer. So the BRCA-mutated ovarian cancer, it works really well, but much less on the breast cancer where BRCA is mutated. Is it the same mutations, or is it the cellular context? It puzzles me because, well, according to our fundamental science, it's just that there are a lot of double-strand breaks.
Yeah, because we do not know exactly. It might also be the tumor type we do not know exactly, actually, and there have been literature about the mutation itself, but that only has been studied in ovarian cancer because those data of tumors were available outside pharma for research. So in breast cancer, they were, so far as I know, not able to do that research yet. And also recently we were at the conference, and there again they discussed this mutation. So in the exon 11, if it's the mutation, then it seems to be predictive. But otherwise, it's the other way around. Prognostic, it's then worse. So that might be a reason that it works better in ovarian cancer than in breast cancer, but we do not fully understand it yet.
Marcel.
Yeah, I also had a question out of sort of general interest on the PARP inhibitor. So there, you said there are many, and are there many because they have different pharmacokinetical features, or is it just because you have different companies that are pushing different initiatives?
Actually, that latest, because the different drugs are from different pharmas, and when you look preclinically, they have been done in vitro studies, and then you see that there is a small difference. So, for example, talazoparib is preclinically strongest. So there is a small difference preclinically, but they work. The mechanism actually that they work is similar. That they work on the single strand.
Yeah, and apparently then the sort of the legality of it is that you can only use those inhibitors of certain tumors because that's been tested, because otherwise you could just use the same maybe inhibitor.
Sorry? Yeah.
Well, why are then some used for certain tumors and others for other tumors? That's because that has been tested.
How exactly. That's how the trials have been done.
Okay, thanks.
Nice.
Yeah, I was wondering, maybe we were talking previous about this this idea of repurposing drugs or using or screening, developing your own drug, basically. I was wondering, is it if there was one of the drugs that Sylvie had a hit on, and it would be a very well tolerated drug, and maybe it would still have a low IC50, but who cares? You just give a very high dose because it's, you know. Would that be something you could test relatively easily if in people who have, for example, recurrence of cancer, and you'd say, well, you know, give it a shot? Or?
Yeah, I think we then need a phase 1 trial. So in a phase 1 trial, I think the first step is indeed the animals, to test it in the animals, and then you go in the patients for whom there's no standard therapy.
Yeah.
So they cannot expect too much from it, and then you do a dose-finding trial. So mostly in cohorts of 3 patients, you start with a relatively low dose, and then you increase per 3 patients. And when the toxicity is in 1 patient, there's a dose-limiting toxicity. Then you go up to 6 patients. If you have 2, then that's the dose-limiting toxicity, and you can't level down. So that's the way it's been tested.
Was there such a drug hit?
I think at the moment there is too much, it's not potent enough yet to think of these things. Yeah.
Thank you. So I think we are at the end of our top research seminar. So I would like to thank the speakers very much for being here, and I hope you also care about fundamental research like we do and enjoy it, but also see the opportunities for healthcare, and go to Susan for all the great technology to make it happen. And of course, if there are more questions, or then please go to research QA LUMC.nl, where you can post questions. But also, if you want to discuss anything else on medical, on research themes, or unrelated to research themes, or research facilities, you can just go there, start a new topic, and then, well, there will people looking out for it and answer your questions. So you can ask questions and find answers, well, basically 24 hours a day, which I think is great. So, and please now join us downstairs for Susan's bitterballen, isn't it? We hope there are bitterballen, but we are not sure.